All right, guys, so last class we had talked about some differences between DNA and replicating itself, and then we also talked about how DNA is transcribed into RNA, specifically mRNA. Now we're going to talk about how, using that mRNA strand, we're able to go through translation and generate proteins based off this. So let's get started. So translation is basically the correct amino acids are brought to the ribosome by transfer RNA, which we talked about last class. So after mRNA is produced, it leaves the nucleus, and this is where translation is going to occur, outside the nucleus in the cytoplasm. Now tRNA is going to, again, be the type of RNA that brings the amino acids to the ribosome at the correct time so that the protein is built properly. And the process of connecting the amino acids into a protein is called translation. You guys can see here, looking at the diagram below, you can see the mRNA strand, which is inside the nucleus, leaving the nucleus. And you can also see the ribosome, which is in blue, reading the mRNA strand. And you can see the tRNA, which basically look like lowercase t's. Those green circles on them are the amino acids. And you can see them bringing these amino acids back to the ribosome. You can also see the long chain of amino acids growing off of that ribosome, and that is called a polypeptide right now. Basically, once the protein is made, it will fold in upon itself in that special, unique shape that proteins have, but until then, we refer to it as a polypeptide. It's kind of like earlier when we were talking about mitosis, when we referred to chromatin and chromosomes as different things, when in reality, they're both the same thing, just a different form. So translation is the process where mRNA is taken to a ribosome and translated into a sequence of amino acids based on its base sequence. So basically the mRNA strand is read 3 nucleotides at a time, and that information is going to tell the ribosome which tRNA molecules should be matching up with that part of the mRNA strand. That will make sure that the appropriate amino acids get brought at the correct time so that we build this protein properly. As I said, guys, every 3 letters of the mRNA strand are read. We call those 3 letters a codon. So looking down at the mRNA strand beneath here, you can see that AUG would be those first 3 letters here. AUG would be the first codon, and in fact, this is called our start codon. So you'll always see that AUG is always the first 3 letters on most of the genes that we're doing. And basically it tells the ribosome that now's the time to initiate this process of building this protein. So looking here, this is called an amino acid wheel, and it might look really overwhelming to you guys. Don't worry, you don't have to memorize it or anything like that, but you do know have to know how to use it. So I'm going to teach you guys how to use it now. So if we're looking at the inner circle of the wheel, that's going to be where we're going to start. So if you remember, AUG, we said, was our start codon. So we're going to use that one for our example. So we're going to start on that inner circle. So A would be in the lower left quadrant. So we see A, so now we're working only in that lower left quadrant for the rest of our codon. So the second letter was U. So you're going to look after A in that lower left quadrant. You'll find the U, which looks like it's pointing like at about 6 o'clock, maybe 5 o'clock from the A. And from there, we're going to go to the third letter under the U quadrant to G. And you're going to see there that there's only one codon that codes for this amino acid, and that amino acid is called methionine. I'll give you guys one more example. So we'll do GAC. So we're going to start again on the innermost part of that circle. So now G is in the upper left quadrant. Then we're going to go again in that upper left quadrant to A. So from G to then A. And then you'll see that C, off of that A, would lead us to aspartic acid. Again, if you guys have questions about how to read these, just let me know in class. One thing I did want to point out is that if you're looking, there are 20 different amino acids. So I could name them all, but I'll let you guys kind of take a look. You won't have to memorize any of these names again. One thing I do want you to know is that AUG is the start codon. I also want you to notice that there are 3 codons that specify stop, and that is going to indicate that the protein is completed and the translation process can stop. So if you look, UAA, UAG, and UGA all lead to stop codons. So again, you will need to know those three. This is just another example of exactly the same wheel that we just looked at. This one's in a square, and I'll teach you guys how to read this one too. Now, I've seen MCAS put both of these on their test before, so I want to make sure that you guys are prepared. So how to use this one? Look all the way to the left, and you're going to notice that the first letter should be all the way to the left. So if we're doing AUG again, we're going to start with A, which is the second row from the bottom. So we know that that row is going to be where we're going to be working. Now the second letter, we're going to go up top. So it's going to determine the column that you're in. And if we look, U was our second letter for AUG, and U is the first all the way to the left column. So if we looked, we're looking again. Left column, second from the bottom, that's where that AU box is. You're going to look, now we just have to find AUG. It's right there, methionine, and again, when you guys are writing these amino acids, please feel free to write just the first three letters. It is perfectly appropriate. You do not need to write the entire thing, and you're going to see that the examples that we use for the rest of today only have the first three letters. All right, so translation, how it happens. Well, the first step of translation is that the transfer RNA, once the mRNA is bound to the ribosome, the transfer RNA that has the complementary anticodon to the first codon on mRNA will base pair with the first codon and bring the first amino acid. So basically, guys, as the ribosome is reading AUG on the mRNA strand, the tRNA molecule that has methionine, the appropriate amino acid, will have the complements of AUG. We already learned that A pairs with U pairs with A, and G pairs with C. So that anticodon would read UAC. And again, this is a way for the tRNA molecule in the ribosome to bring the appropriate amino acid at the correct time, because this is very crucial when we're building these proteins. We're going to talk about later today what happens when these mechanisms don't work perfectly. Now, once this is done, the ribosome slides to the next codon, and a new tRNA molecule with the correct anticodon will bring another amino acid. So you're going to see here now we have two tRNA molecules in the ribosome at once. And 2 is actually the most that can fit in the ribosome at once. There is a third slot that the tRNA molecule will slide to, but that is right when it's going to be exiting the ribosome. In fact, we're going to get there on the next slide. So an enzyme then joins the amino acids together with a peptide bond to start making the protein. If you guys remember from earlier this year, we talked about how amino acids form very special bonds between these amino acids called peptide bonds. And again, we won't get into any further detail on that, so you guys can just understand that it's a peptide bond. But again, these peptide bonds will form between the two amino acids once we have 2 there. Now that the first tRNA molecule has had its amino acid bonded with the other amino acid, it now no longer has one, and that tRNA molecule will be released. The mRNA will then slide to the next codon, and the process is going to continue, adding more and more amino acids and bonding them to this polypeptide chain until eventually it reaches one of those stop codons. And again, those 3 were UAA, UGA, and UAG. And what that means is that we have completed making the protein. And on that note, the ribosome will then release the protein and let it go out into the cell. And that protein, again, that polypeptide really at that moment is going to first fold in upon itself, creating that very unique shape of that specific protein. And then it is going to go off and do whatever proteins do. And some of those functions of what proteins might do, we've talked about earlier this year that proteins are important for enzymes. Enzymes are going to break down and help initiate chemical reactions, and really they help with pretty much any chemical reaction that occurs in your body. They also help with the structure of cells, so they provide structure on a cellular level as well as an organism level. They help to transport molecules across the cell membrane, specifically with active transport when the concentration gradient goes from low to high. We learned about how they are used to make antibodies, and antibodies help us to fight off viruses and disease. Metabolism, they also help to break things down, whether it's digestive enzymes and digestion, or other metabolisms such as a cellular level when we're talking about cellular respiration. And lastly, they also help to repair body tissues as well. Now guys, on your test you will be asked what are some functions of proteins. You can give me any of these. The three really important ones: enzymes, transport, metabolism. Those are really important ones. Here's a diagram showing you the stages of translation, and I know later today I'm going to be showing you guys a video, so please check out those as well. Whatever works for you to best understand this process. I know it's a pretty complicated one, but again, whatever helps you guys. So looking at this diagram, you can see on the top here we have the ribosome that is bonded to the mRNA strand, and it's reading the very first codon, AUG. You can see that the first tRNA molecule has brought the amino acid there, and in the next diagram you can see that more tRNA molecules continue to bring the amino acids in, and those amino acids are bonded together. Also note that only two tRNA molecules fit in the ribosome at one time. Lastly guys, in the last picture you can see that the ribosome has now reached a stop codon, which is going to determine that the protein has completed. And you can see that the protein now, the polypeptide, has folded upon itself, becoming a protein, and it will now go to complete whatever it needs to in the cell. So next we're going to talk about mutations. And like we said, sometimes this genetic code, when we're copying it in transcription or even in DNA replication, there might be some changes that are made. And sometimes, again, those mistakes are just due to replication. The cells do have mechanisms to kind of proofread it, but sometimes those mechanisms don't work 100%. And we also talked about how exposure to radiation and exposure to chemicals could also either prevent those mechanisms from happening or alter our DNA. Now there are three different types of mutations that we're going to talk about today. We have substitution, insertion, and deletion. And one of those, substitution, is known as a point mutation. And point mutations involve only one or a few nucleotides, and they occur at a single point in the DNA sequence. Now, substitution mutations are much less invasive than insertion and deletion mutations, and here's why. When we talk about a substitution mutation, we're talking about just one base being changed to a different base. This is going to affect really at most just one amino acid. If we look down here at the diagram, you can see first on the left the original DNA strand, and on the right you'll see where the mutation has occurred in red. Now you're going to notice that here this C has been mutated into a T nucleotide. Now that's going to make a difference when we go to create the mRNA strand and eventually our amino acids in our protein. So if we look, the amino acid sequence on the original strand was methionine, arginine, threonine, and phenylalanine. Well, if we look at the substitution mutation where that has occurred, you're going to notice that it's only affected one amino acid, histidine. So again, substitution mutations, far less invasive. Insertion mutations, on the other hand, are far more invasive, and that's because when just one base is inserted, it shifts the genetic code down by one. And remember that because codons are read every 3 letters at a time, this is going to change every combination of letters after that point of that mutation. So if we look on the before, we already talked about the order of those amino acids. We notice here with the mutation of insertion that not only was that second amino acid affected, but every amino acid after that no longer matches the original strand. So this protein likely is not going to function properly. If we look with deletions works in exactly the same way, but the opposite. Now instead of adding a base, one base might be left out. And that would, again, result in all of these, the entire code, the mRNA strand, shifting down one. And that's going to completely change the combinations of codons that we're going to have. And if you look here under the deletion protein that's being built, you're going to notice that not only were the next 2 amino acids incorrect, but that last one, codon, it only has 2 bases. It doesn't even code for an amino acid. So that protein will definitely not function properly. So we've kind of already talked about it, but insertion and deletion mutations are referred to as frameshift mutations, and that's because they change the entire reading frame of the mRNA, because all of the nucleotides after it are going to shift down one. So guys, the last thing I wanted to mention today is that we also could have chromosomal mutations, and chromosomal mutations involve changes of either the number or the structure of the chromosome. So we mentioned trisomy 21 would be in addition of one chromosome. Well, sometimes you could have the deletion, duplication, inversion, or translocation of part or an entire chromosome as well.